All right, thank you. All right. Good morning again, everyone. Welcome to the chromosome recognition lecture. We're going to be talking about structurally normal G-banded chromosomes today, and we're going to talk about how to distinguish a 1 from a 2 from a 3, etc., and a little bit about how we actually construct a karyotype and some of the things to look for. So, like I said earlier, please feel free to stop me in the middle and we will get going. So I don't know what the background is for Eric, Casey, and Matthew. So I'm just going to go very basic. And so this is a normal G-banded karyogram or karyotype. You will hear me say karyotype. In the cytogenetics world, there is an argument going on about what we should actually call this, a karyogram or a karyotype. But the old term is karyotype, and I've been around for a long time, so that's usually what you're going to hear me say. G-banded stands for the way that we actually get those bands. It's a Giemsa-Tripson technique that we'll talk about in a whole nother lecture, but this is a G-banded karyotype. And you can see that there are, it's just an orderly arrangement of the chromosomes, the 1s, the 2s, the 3s, the 4s, 5s, and down to X and Y. If this were a female, of course, there would be 2 X's and no Y. But we're going to be talking a little bit about terminology today. So there are 22 pair of autosomes, which includes chromosomes 1 through 22. And those are going to be the same in males and females. But then the sex chromosomes, XY for male, XX for female. And you can see, even if you can't recognize the chromosomes yet, you can still see that there are a series of dark and light and kind of gray bands up and down the chromosome arms. So when we talk about metaphase chromosomes, so when we look at chromosomes, we are looking at chromosomes that we have arrested the cell during cell division. And again, we'll talk about all of that technology a little later on in boot camp. But we are looking at metaphase chromosomes. And there are 3 basic sizes, or 3 basic centromere positions. There's metacentric, which the centromere pretty much divides the chromosome between long and short arm. Metacentric, the P and the Q arm are going to be roughly the same size, so short and long arm. Submetacentric, the centromere is closer to one end, and then there's a special group of chromosomes, the acrocentric chromosomes, in which the centromere is extremely close to one end, but the I guess identifying part about the acrocentric chromosomes is they have stalks and satellites. Okay, so terminology to P-arm is it's also called, you'll hear it's called the short arm. P stands for petite. So when the international group was naming all these conventions, the French kind of won out on that. Petite in French is small or short. And then the long arm was named Q simply because Q comes after P in the alphabet. So no more scientific than that. I guess the other thing to see, telomeres, we'll talk more about that. And then on a metaphase chromosome, so the chromosome's already replicated, the DNA's already replicated, getting ready for cell division to form 2 different cells or 2 daughter cells. And at that point, we talk about sister chromatids. So again, the chromatin has replicated, so each chromosome has really 2 P arms, 2 Q arms joined together at the centromere. And then once cell division occurs, or once the chromosomes start separating, then there will be a break, and this will be 1, 1, this will be 1, and go to the different cells. The other thing to point out on here are the stalk regions. These code for ribosomal RNA. So they're the site of the nuclear organizer regions, which again comes in more when we're talking about cell division. The satellites are highly repetitive DNA, but both of these regions can be highly polymorphic. And by polymorphic, I just mean that there can be different shapes, sizes, staining intensities, lengths. And that really doesn't have a whole lot of clinical consequence, but it's certainly it's a region that can be unstable when we start talking about structural chromosome abnormalities. So the other thing that you will hear cytogeneticists talk about is an A group, a D group, a G group chromosome. And I wanted to show you where that terminology came from. So this is a paper from 1962 that shows a routinely stained, or it's basically Giemsa. There was no we didn't know how to do banding at that time. So the chromosomes were just stained with Giemsa. So they're, the same. You can't see any bands because there are not any there. And it's kind of hard to tell, especially in this group, one from another. So by convention, the people that were, again, the international community decided that the A group chromosomes would be chromosomes 1, 2, and 3. And you can see that there are different centromere positions for 1, 2, and 3. That B group would be 4 and 5. And C group, the X chromosome is included in the C group. And then the Y chromosome, this is a female karyotype, the Y chromosome would be included in the G group chromosome. So D group acrocentric, G group acrocentric. But this is what they had to work with until about 1972, when they started being able to figure out different ways to band the chromosomes and identify them more closely. So again, some cytogenetic terminology that you'll hear in the cyto lab a lot: proximal and distal. Proximal being closer to the centromere, distal being more toward the end of the chromosomes. Each pair has 2. We get one number 1 from mom, one number 1 from dad, and those are called homologs or homologous chromosomes. So they're members of one pair all the way through. Band resolution, we'll talk a lot about that in a bit, but that's a number of bands per haploid set of chromosomes. So when we're looking at a cell, it's got of a normal male or female, we've got 46 chromosomes. The band resolution is based on a haploid set, so on 23 of those chromosomes. We talked about, I talked a little bit about polymorphisms, heteromorphisms. And like I said earlier, those are not generally associated with the phenotypic or reproductive consequences, but they can certainly play an important role in chromosome breakage. The other thing that we have also come to accept or realize, appreciate, is that there are euchromatic variants. So there are variants that are not considered heteromorphisms, but because they're in the G-banded regions. But if there's some gain and loss there, it's, again, usually no phenotypic or reproductive consequences. Now, we'll have a whole nother lecture on ISCN, but I needed to kind of introduce it today. So the International System of Human Cytogenomic Nomenclature. And what that does is just to facilitate communication of cytogenetic test results between labs, between different groups, different doctors, et cetera. In the early days of clinical cytogenetics, I think each lab had their own system of how to name what an abnormality they were seeing. So it became apparent very early on that there needed to be an international system. And so the people, the cytogeneticists in the field at that time got together, and there's a whole book that actually ISCN 2020 that you will need to purchase because there are a lot of rules and regulations. I will give you a talk on it, but I even refer to it. I referred to it yesterday to make sure that I was putting something correctly in the nomenclature. So I guess the most basic thing to know today is that the correct nomenclature for a normal female is 46, XX. So this is how you will see that written on a report. If it's a normal male, you'll see 46, XY. Now, when we're going to be talking a lot about bands today, and each of those bands will have numbers and letters signifying what they are. So a major band can be subdivided into a higher resolution as we get to the higher resolution chromosomes. 500, this is about a 500 to 550 band level from the ISCN ideogram book. Now, the earlier that we catch a cell in metaphase or even at the end of prophase, the higher the resolution is going to be. And that also we'll talk more about that as you get further into boot camp about how we do that in the lab. So let's just, what I've given you here are from the ISCN book, and this is a 400 band level ideogram, 550 and 700. Now, usually we're with constitutional blood chromosomes, we're working somewhere 550 to 600. With the oncology chromosomes that you'll see much later, we're working more at the 400 band level. So how do we identify a one from a two from a three? First of all, one is the largest chromosome, and it's got, at the 400 band level, it's got two major, what we call landmark dark bands. This is in the proximal region of 1P. These then begin to split into a higher, at higher resolution, they split into really 4 bands. So if I'm looking at these 2 bands on a 400 band level, I'm not going to see the split. I'm not going to see this lighter band in there. But when I get, these are 550 to 600, I see this band split. This is this band, and it's split here. And this is the region in here. And then that will be this 31.3 band. The quality of the banding has a lot to do with the resolution of the chromosomes. You can have really nice long chromosomes, but if your banding procedure does not work well, then you're not going to have all of these subtle little gray bands. It's going to be more of a smear. So that's what we constantly work with the technologists to get the banding as what we call as sharp as possible. But you can see this 400 level and then again down to 600 level. So when I look at a 1, how do I identify that? I'm looking for really at the end of 1P, a large light area. It does have dark bands in there, as you can sort of appreciate here, but it's certainly looks much lighter than overall than this region. And then in the Q arm, there are 3 major dark bands here. So this, and this one. Yeah, these obviously are much darker than this. And if you get over here a little bit longer, that is about, and again, a better banding quality, that is about the same darkness as those. But you're going to be looking for 3 major dark banded areas in the Q arm. Now the chromosome 1 is one that has heterochromatic region just under the centromere. This is actually heterochromatin right in here. Right here, you see, this is all heterochromatin. The 2-2 band actually doesn't start to about right here. So these, you can, again, I think maybe from this, appreciate the size difference, or even from here, we've got kind of a doublet in the heterochromatin region and a singlet here. Again, no clinical consequences associated with that. So 2 is usually about the same size as chromosome 1, but the centromere is appreciably in a different position. This is more of a submetacentric chromosome. And the 2P has 4 major dark bands that you can see on the ideograms here. And we can see that here. Again, as we get longer, you get more bands in there. So you're going to be starting to identify chromosomes with the shorter ones. They'll be a little bit easier, and we'll work you up to the ones with the more bands that kind of confound some things. But the main thing to look for with chromosome 2 are those 4 major dark bands in the P arm. And then in the Q arm, when I'm reviewing a case and looking at chromosome 2, I'm concentrating on 3 regions in the Q arm. It's this kind of lighter region up here, and then a dark region, there's a lighter gap, and then again, 3 dark bands, obviously separated by lighter bands. So the distal third has major dark bands and 2 thinner dark bands here. The middle third, major dark bands, the upper third, a little bit lighter. And I know this can look pretty confusing right now, but you will get used to it, and it's just going to take a little bit of time. Now, chromosome 3 is metacentric. So the centromere pretty much separates a P and a Q arm that are almost the same length. The one way to really recognize chromosome 3 is to look for that dark cap. This is one of the few chromosomes that has a really dark cap at the end. Now, if you get long enough chromosomes, you can start to see this little gap. It's called band 26.2. You can start to see that, but for the most part, you're going to be seeing a chromosome that has a dark G-band dark cap on it. Then to look kind of down the chromosome arm, P arm, we've got major dark bands, we've got a gap, we've got other major dark bands and centromere, and then a gap in the middle, kind of in the middle of the Q arm, a little bit toward the centromere. But you've got, this chromosome actually has 2 recognizable lightly stained areas on either side of the centromere, and then they're flanked by darker staining regions. And the distal half of the Q arm, it's got from anywhere from 2 to 5 or more dark bands, depending on the resolution. I know this 400 band ideogram is showing 3 dark bands in here. If we get some of those really, really short oncology chromosomes, you may just see a couple. This actually confused into each other. But like I said, when I look at 3s, I'm looking for that dark cap. I'm looking for the lighter area. Then I'm looking for a gap that separates 2 dark band regions, and then in the Q arm, a gap that separates the proximal from the distal dark region. So that's our A group chromosomes. And here's just a comparison of, chromosomes 1 and 3 are metacentric, 2 is submetacentric, but that will give you, kind of a comparison to use when you're beginning to mark your chromosomes on the cells that Nicole will be giving you after this lecture. Okay, so chromosome 4 and 5. Chromosome 4, the P arm has 2 dark bands in its center, but it has a very light cap, usually quite large light cap. Then the centromere, and then we've got a dark band at just under the centromere and light area. There's a series of dark bands. These chromosome 4 will be one that can cause problems because it is one of the chromosomes that doesn't band super well. You've got to have a really good banding procedure to get these bands to be distinct like you're seeing here. So sometimes this region can kind of look like a smear, kind of like here. So, but again, with chromosome 4, we're looking at a large lightly stained area on the P, and then we were looking for a doublet at the bottom of the Q arm, and we're looking for this. Some people have actually called these shoulder pads. The 4Q has shoulder pads, and that is separate from, that's a little bit different from chromosome 5, which also has the dark band in the middle of the Q arm. But chromosome 5 has a series of 3 dark bands with, as you separate out. Chromosome 4 has the more of 4 dark bands to a series of 4 to look at.
Dr. Smith.
Yes.
Can I ask a question?
Sure.
I was just wondering, what did you mean by shoulder pads? I think I missed.
Okay. Okay. So, so you're way too young to have worn shoulder pads and everything you wore. So it's just basically something at the top of the shoulder of the long arm. So here's the centromere right here, and then this really dark band is it's distinct. It stands out. And
I see what you mean. Right below the centromere.
Right.
I see. Okay, thanks.
Oh, you're welcome. No, I'm sorry. I'm using terminology that you're way too young to understand, to have known. Sorry. So let's kind of go on that with chromosome 5. You're saying, well, there's a dark band there. Yes, but it's not nearly as distinct as the one on chromosome 4. Okay. So with 5, we've also got a lighter area at the top, especially on the shorter chromosomes, but you get any kind of length on 5, and 5p definitely looks different from 4p. You've got this region up in here that it usually not as dark. This one, the staining does look like it's just as dark as this, but often this band is it's dark, but it's a little bit lighter than that. So again, with 5, remember, much lighter region until we get to that series of 3 dark bands. And then yes, you've got a doublet maybe at the bottom, but it's usually separated. It doesn't look quite like the 4s. So let's go to the B group comparison and see 4 and 5. And again, these are fairly long chromosomes. And I can see you're going to probably mix up 4s and 5s in the beginning, and it's certainly understandable when they're this long. I think the shorter ones are a little bit easier to tell, but a bit darker and heavier here than here. 1, 2, 3, 4 bands there. This is the same band split, 1, 2, 3, and then this region looks pretty similar. And sometimes if you're looking just at this region sometimes on a poorly banded, it can look kind of the same, especially if you don't have this band in here. But for 4s, it's just more of a doublet, and you really probably wouldn't even think of a doublet for this region of 5. So,
excuse me.
Any questions?
So how to find the dark color near the centromere for chromosome 4 from this figure?
How do we find the centromere?
The dark color.
I'm not sure I know exactly what you're asking, but the centromere and on the higher resolution, thinner chromosomes, sometimes it is hard to see where there's a constriction, but you definitely want to look for the constriction. And as you look at more chromosomes, it's going to be fairly obvious where it is. But this is the centromere right here for 4, and this is the centromere right here for 5. So you can see on these, you can kind of see a gap. Yeah. So
I think Eric may be trying to find the shoulder pad on the chromosome.
Okay.
Right.
Okay. So on 5, there's you usually won't hear people talk about shoulder pads on 5.
On 4.
On 4, it's right here.
4, yeah, near the centromere.
Right near the centromere, just under the centromere. So here's the centromere right here, and then there's a white band, and then this is what I was talking about. This is a major dark band that you don't have on chromosome 5. It's just more of a kind of a even, staining on 5.
Oh, I thought it was very close, but it's not that close to the symptom.
Well, it on the higher resolution, no, you're right. That's a good pickup there. So on these shorter chromosomes, yeah, it is pretty close to the centromere, but there is something in there. This actually is a good time to point out euchromatic variant, because we do know that 4q proximal light band right under the centromere can vary in length between individuals. So this is one of the regions of a euchromatic variant. So in some fours, it's going to look like there's more of a gap there between the centromere and that shoulder pad than it will in other chromosomes. But the resolution will play a role in that as well.
Thank you.
Does that help? Okay.
Yeah. Thank you.
Okay, sure. All right. Okay, now we get to the C group chromosomes, which you'll have a lot of fun with. So we've got chromosomes 6 through 12 and the X. All of similar in size. 6 is the largest, 12 the smallest. X is probably somewhere in the 6, 7, 8 chromosome size range. So this will be probably the group that gives you the most problems. So with 6, what do we look for? Even on the shorter or longer ones, we're going to be looking at, this is a submetacentric chromosome, so the centromere is right here. We're going to be looking for a gap, a light staining gap in the pretty much in the middle to proximal region of 6p. Okay, and then the Q arm has a lot of bands in it. So really the best way to start recognizing chromosome 6 is with the P arm, with that lightly staining gap in there. Then you're going to get a series of areas. So right under the centromere, we've got, again, you could call it a shoulder pad, but a darkly stained area. Here's a dark band, this doublet here, which does not separate at the lower resolutions. And then this region you will find doesn't always band very well either. So it may kind of look like a smear on electrophoresis gel sometimes. And then, but the Q arm, we definitely want to see these 3 distinct dark bands. All right. Now, chromosome 7 also has a kind of a major area on the P arm that is more lightly stained. But remember the cap on the on chromosome 3? Well, chromosome 7 has a light cap, but it also has a very dark staining area right under that light cap. And you're not going to mix up a 3 and a 7 because they're so different in size, but it's kind of something to refer back to. So we've got, again, the light cap, a very dark band, a lighter region to the centromere. And then in 7q, we've got really 2 major dark banded regions that will split into various bands. And then sometimes a 7q the end of 7q, this band may remind you of the distal end of 5, but again, you're not going to mix up a 5 and a 7 because of the difference in size. And it is a little bit different, but I can see how some people, if you were just looking at that, would think 5q. So let's keep going, and then we'll have that comparison screen. Chromosome 8 has also a gap on the shorter ones in white area in the P arm, and when we put them all together, I think you'll see how it's different. 8q, again, a lot of bands here. And at the higher resolution, they're going to start splitting. But if you're looking, so this week, next week, when you're working on the shorter chromosomes, with 8, you want to look for, this also has a dark cap on it, but it's not quite as dark as 3P. And this will help you distinguish 7 and 8 because yes, there's a major dark band here, but that does not look like 7, okay? And then in the Q arm, we've got a region that's, there are some dark bands in there, but it's not nearly as darkly stained as this region and this band right here. And then 8Q ter should be extremely light. You'll hear some of the technologists talk about a fluffy area on the chromosomes, and when they say that, when you hear that, they're simply meaning a very lightly stained area. So now chromosome 9 it also has 2 dark bands in the P arm, but they're closer together than the dark arm that's on the 8. Especially when you see the karyotype all together. There are 2 sets of dark bands in the Q arm, and they also split as they get higher resolution, like we have here. This region is another one of our heterochromatic regions. So we talked about 1Q proximal right under the centromere, and now chromosome number 9 has the same light. With 1, we saw dark bands in there that were heterochromatin. The 9 has lightly staining heterochromatin on the G-banded karyotypes. So this again is very polymorphic. It's what we call C-band positive, and we will talk about different banding patterns in a later talk. So just kind of tuck this to the back of your mind right now. That doesn't really help you identify the chromosomes. This region, though, is really prone to inversions. And an inversion is when you get a break, 2 breaks in the same chromosome, and that middle segment reverses its orientation. So this is an inverted 9 down here. What has happened is there's been a break right above the centromere in this P13 region, or actually right at the interface of P12, P13, and then a break at Q13. And then all of this is flipped up, this is flipped down, and that's what you're seeing here. So, this is the one inversion, actually, I shouldn't say just one. There are several inversions that are right around the centromere on various chromosomes that are known polymorphisms. Again, no reproductive or clinical consequences from this. So you will see as you get into the lab and start looking at cases and seeing reports, you'll see, let's say, this was a normal male or normal female karyotype with a paracentric inversion of chromosome 9 present as a normal variant. And if we see this in a bone marrow, we would say normal constitutional variant. But this occurs in more than 1% of the general population, so you will definitely run across these in some of the cases that you do. All right, chromosome 10. The P arm's doesn't stand out a whole lot. There's a dark band right in the middle, but the Q arm really does. There are 3 major dark band regions, all fairly equally spaced along the Q arm. And of course, they split as the resolution increases. And on the shorter 10s, you're not going to see this dark band down here, but that's certainly something on constitutional cases that the 10s, we need a good look at those. This is a region that is known to be deleted in some children with dysmorphic features, intellectual disability, et cetera. So it's a syndromic deletion, but this is one region that we really look for when we're looking at that. But these 2 bands right here equal that one. And then this one, you're beginning to be able to see right there, these bands in here, and then this group of bands represent this part of the 10. So. Okay, now 11s and 12s are often easily mixed up. But what you want to look for with chromosome 11 is a dark band, 2 dark bands, a doublet that's close to the centromere. And then in general, you've got a lighter region on the for the distal half of the P arm. And you can, again, as you get longer, they're going to separate out more. But the main thing to look for with 11 is that dark region just above the centromere. Then we've got a very light region just under the centromere, and then 2 dark bands that on the higher resolution chromosomes will divide out into 4, 5, 6 bands even. But this very dark region in the middle of the Q arm, and then the dark region at the proximal P arm, or how to separate an 11 from a 12, which also has a very dark region on the P arm. The P for 12 tends to be a little bit shorter than the P arm for 11. And then your dark concentration of dark bands is higher up, closer to the centromere, and there's a lot more of the lighter staining area at the bottom, like here. And yes, remember that in any lightly staining area on a chromosome of this resolution, you get up to 650, 700, 800 bands, and you're going to see dark bands. Okay, and then the X. X is often misplaced, especially when you're just learning your chromosomes. So, expect that to happen, but I think you'll get to where it will be fine. You'll be able to recognize an X from an 8. Often I see an X misplaced, it's turned upside down, and because this actually does can, especially on some banded preps that are not great, it can look a little bit like 8P. So be aware of your X's and 8s, but if the X is oriented correctly, and if you can see the centromere, which is right here, then on this chromosome, you know that this is the P arm, and the XP really doesn't look a whole lot like XQ, like 8P, sorry. So with the X chromosome, you'll see on the karyotypes that we do, we put X and Y at the end. You may see some illustrations in papers, or in another lab, the X chromosome would be on the karyogram grouped as a C group because of its size. So at the lower resolution, X has the one major dark band in the P arm, and a major dark band under the centromere, not right next to it, but with some white gap. And then you've got a series of dark bands. But these are the 2 landmark bands to look for in X. And just then again, as you get to the longer ones, you're going to have to kind of change your focus and think, yeah, okay, that's going to separate out, but it's still a fairly, it's a series of dark bands. So here's our C group comparison. All are submetacentric, but and like I said, most likely be confused. Okay, so again, I think if you can use this slide as when you're beginning to mark your cases and do some karyotyping, to kind of see the differences of the we've talked about that light gap in 6P, the light in 7, but the distal end doesn't look the same. Your long arms certainly don't look the same. Here's that dark band at the end of 8P with a gap, and then a lighter area, another large gap, but it doesn't look like that's a little bit different from 6. And again, your Q arm is going to be different. So, and then 11, sorry, that was a very bad 12 to put in there, but at least this one looks okay. but you can see how, yeah, there's how much bigger the 11P is than the 12P. And then in the Q arms, you've got the group of dark bands a little bit closer to the telomere than you do on 12. Okay, so we're halfway through. So chromosomes 13, 14, and 15, these are the one group of acrocentric chromosomes with that variable P and Q arm, variable P arm, stalk, and satellite. And I've shown you a couple of examples here that this is actually probably a double stalk. So very extended stalk length, the difference in staining intensity of the short arm, of the satellite versus this one. Sometimes the satellites look a little, they kind of fade out or just look a little runny, smeared. But for chromosome 13, we've got major dark bands in the middle and toward the end of, so this series of dark bands is down on chromosome 13. Obviously this is a dark band, but you've got the lighter region at the top. The middle dark band is going to be one of the first ones to split on chromosome 13 as you get a higher resolution. Now 14 has that gap in the middle. So we've got major dark bands on near the centromere and major dark band toward the telomere, but that's again has a wide gap of lightly stained or not so darkly stained area on 14. Whereas 15, it's got that, again, a major dark band in the middle of the Q arm, kind of like chromosome 13, but the difference, I think it's easier to see on this next one. When you've got that major dark band on 13, you're going to expect to see at least two more dark bands. When you've got that major dark band on 15, you're going to expect to see a lot more lightly staining area until you get to the very terminal end. And then 14 is fairly easily recognizable because of the major split. So I think I covered everything on that, yes. Okay, so then chromosomes 16, 17, and 18 make up the E group chromosomes, and they tend to be much more metacentric than the C groups. The chromosome 16 also has a heterochromatic region just under the centromere, and you can see it right there and right there. So with chromosome 1, we had some darkly staining heteromorphic regions. With 16, we've got some darkly staining, and with 9, it was a light staining. But all of those would be C-band positive. And again, we'll talk about C-bands a little later into boot camp. So for the P arm, a lot of times actually the thing that I guess stands out about the P arm is, again, how much of a smear it can look like. There is that dark band at the telomere, or not at the telomere, but the dark and light band at the telomere. But a lot of this this is a region that does not always cooperate in the banding. But we usually are able to appreciate two major dark bands in 16q. And those would be here and here on this one. This is the heterochromatin region. So this band corresponds to that, and that band corresponds to that one. Now you see, as we're getting shorter chromosomes, we're getting fewer bands to have to deal with, that will help us identify it. So again, chromosome 17 is more is submetacentric. Actually, I think I misspoke earlier. This group has pretty good representation of metacentric and submetacentric. Not all of them are metacentric. Again, with chromosome 17, there's one major dark band in the P arm, but look at 16, it's the major dark band is more toward the end of the chromosome, and with 17, it's actually either in the middle or more toward the centromere. It can appear more toward the centromere, again, depending on the length of the chromosome. And then the gap in 17q is followed by a doublet, and we do usually talk about this doublet on 17q. So submetacentric, major dark band, a large light area with the doublet toward the end. And then chromosome 18, which is the last of this group, obviously very submetacentric, but the P arm is usually very pale, and it can have a thin dark cap on it. Sometimes it looks a little darker like that, but for the most part, you're going to be looking for a submetacentric chromosome for 18 that has a good bit of light area, and then has a series of usually four bands. Sometimes it will look like 3 on the shorter chromosomes, but certainly on the routine ones that we use for our blood studies, or the high res we use for our blood studies, we should be seeing at least 4 dark bands. You can think of this as a set of 2, followed by a lightly stained area, followed by a set of 2 dark bands, and then the lighter telomere. So here's our E group comparison. And I think you can again see, maybe appreciate the kind of smeariness of the banding there. The fact that 17 has a very dark, distinct band right cutting it in the middle, and then the 18 is pretty lightly staining, you know. And then with the P arm, with the Q arms, you've obviously got the series of 4, you've got this doublet, and then you could call this a doublet, but they're definitely placed differently on the Q arm for 16 and 17. Okay, then we got 2 more groups to go here. So chromosome 19 is metacentric. It has a very prominent centromere, and it is mostly really lightly staining. There are dark bands in it, but they certainly do not stain as darkly as some of the ones we talked about. So we've got a kind of a doublet here on the higher resolution. On the lower resolution, you're just going to see 1 dark band, but I use dark in relative terms here. And then again, your 19q is going to have, again, another dark band. It's a little bit darker at the telomere than the P arm. So it's really easy to miss karyotype 19s in that they get turned upside down. And I have to get on to even our senior tech sometimes will turn in karyotypes with it 19 upside down, and they know better. But it's just when you're going through, it's really easy to flip the chromosome and have the Q arm at the top, which obviously is wrong. So, but I think the longer 19s, you should not have as much trouble making sure the orientation is correct. And then chromosome 20 is about the same size as 19, but it's got darker bands in it. So the P arm has a darker band and 2 dark bands in the Q arm, and they're fairly evenly spaced. Sometimes the 20 can look like a small 16q down here, especially on a very low resolution. So you just have to be sure that you put the larger ones in the 16 slot and make sure that your 20s are actually the 20s. And then so here's a comparison of the 2. You see, lightly stained chromosome, this one has bands. And I guess the other thing to point out is 19 usually has a very prominent centromere, partially because the arms are more lightly staining, but in general, 20 definitely does not have as prominent a centromere region as 19 does. And then chromosome 21 and 22 are other acrocentric chromosomes, again, highly variable regions. And then the 21 is, there's a major dark band in the proximal Q arm. Oh, the other thing I should point out with the acrocentric chromosomes is a lot of times you're not really going to be able to see this P arm. It kind of all blends into the stalks and satellites or into the centromere, because these are extremely tiny. So anyway, with the 21 major dark band and the proximal Q arm, but also note that there is the light band between the centromere and Q21 that can be seen on some of the higher resolution chromosomes, like right there. You won't see that all the time. So mainly what you need to look for is that 21 has a dark band that's very close to the centromere, and then the rest of the chromosome is a lightly stained region. Unlike 22, that has 22 looks a little bit more like 19 in the fact that it stains lightly most of the up and down. It's got a very prominent centromere, and the staining in the long arm is a lot less distinct or dark than the like 21 or the 20, but 22 has a major dark band right in the middle, which you can see here. Okay. So the other, I guess the other, what I was going to say also is sometimes these are not particularly long stalks and satellites, but I have seen long stalks and satellites, so much so that I sit there and go, is that a 19 or is it a 22? So there are other ways to tell as you get a little bit more into this about which is which, but I have seen miskaryotyping due to some really large stalked satellites in that region for 22. And then the Y chromosome, submetacentric, again, is placed with this group because of its size, but it has a dark cap on the P arm. It has the proximal Q arm has 1 to 2 very indistinct bands, and then this is also heterochromatin. So again, this would be C-band positive. This varies. Most of the time when you see a difference in length on the Y chromosome, it's because of this region right here. And you can see it does not G-band well. So you will be seeing some more examples of structural abnormalities of chromosome Y and we will talk later in boot camp or in the cyto rotation about what we can do to figure out exactly what's going on with the Y with various staining techniques and FISH techniques. But just remember, there is a large variability in the length of that heterochromatin region. And so here's our G group comparison with the landmark band on 21 and the lighter landmark band on 22 in the middle, and then the Y chromosome. Okay, so I've talked about band resolution, band lengths. Wanted to take the last few minutes to briefly go over a couple of different methods of counting bands, because we don't want to sit there. I said the high resolution is dependent on the number of bands in a haploid set of chromosome. We don't want to count every band between chromosome 1 and 22 and 1 through 22 in X. So this is a Vancouver method named because it was developed there. And what they do is count the G positive bands on these chromosomes. So like 1P, 3132, which I've marked for you, 11P and 12Q, and then both arms of 10 and X. So they count all the G band positive or G positive bands and do not count the centromeres. And then you extrapolate from there. So if you had a karyotype that had 18 bands in these regions, then you would say it was at the 350 band level. If you had 40 bands, you would say it's a 550. So we do an extrapolation from looking at those regions. Now, the one that I use mostly, because I only have to count the bands on one chromosome, is the Johnson and Stoller method. And we count the light and dark bands on 10p and 10q. You also count the telomeres and you count the centromeres. And this is accurate within 25 bands. It's been shown over and over again that it's actually a very accurate method. So again, if by counting all of this on a chromosome 10, I've got 15 bands, then I'm talking about a 425 band level resolution. If I've got 30 bands, I've got a 600 band resolution. So those are some of the ways. You don't need to worry about that this week, but I just wanted to go ahead and give you that information so that when you do start working on cases, and get a little bit more into identifying chromosomes, you can give us an idea of what you think the band resolution is. And then the last 3 slides, I wanted to just kind of talk about karyotype in a cell. So you, after this today, you need to, if you're on site, you need to come on site at least and get from Nicole a set of metaphase cells that you will then start trying to, you're just going to label them. You're going to identify the 1s, the 2s, etc. Eventually, after you get some experience with doing that, then you will be learning to karyotype or karyotyping cells. And certainly when you start your logbook cases, you will have to do the karyotyping. So I just wanted to kind of point out some things on what the best practices, I guess, for karyotyping. So the best practice is when you see, this is a metaphase cell. So you've seen, we've been showing, shown karyotype. This is what that karyotype, comes from, is a cell with the chromosomes are all spread out. So I like to divide the cell, this particular cell, I divided into 3 sections, counted the chromosomes in each section, and I end up with 46 chromosomes, which is what I was expecting. If you're karyotyping a cell that somebody has already done the analysis on at the scope or even on ASI, then you want to check what your counts against what they got. And if there's any discrepancy, you want to figure it out right then. So when cutting the chromosomes out of the metaphase, we want to trim evenly around the chromosomes as much as possible. We don't want to trim right up to the edge because it makes analysis much harder. But we also don't want to leave too much white space around them. And I've got it, I'll show you in a second. ASI, which is our karyotyping system, it tends to cut off satellites, which I've arrowed here. These would have been cut off. So it would have only picked up this chromosome. You have to go back and include those satellites. It also sometimes cuts off pale terminal ends. The other thing I've seen it do is separate chromosome 1 into 2 because it thinks that that's a gap there and that's a different chromosome. So when we're trimming the chromosomes that are crossed over, we're not going to karyotype it in the crossover. We have to get that one and we have to get that one. And if you were doing this with paper and scissors, you would obviously need 2 copies of the cell to get these 2 chromosomes. Fortunately, ASI makes it a little bit easier than that. You can tell ASI, I want this chromosome cut and I want this chromosome cut. So again, in the overlap, you've got the tip of, and you've actually got the tip of 4P touching 1Q TER. So you're going to leave a little bit of space or you're going to get a little bit of that. If you can, mainly, so when somebody's looking at a karyotype, they can tell, okay, that was up against something. Sometimes it can distort the chromosome. This one's not really distorted, but in others it can. And we need to see when we're analyzing that karyotype to send out a report, we need to see if is that a distortion because of where it was placed in the met, or is there something really wrong and we need to look further. So, so this kind of just shows, this is, these chromosomes were not trimmed closely enough. This is a fairly good trim here. The chromosomes that this is trimmed too much, you need to come on down because there are satellites under there. So basically, you'll get used to all of this, but not trimmed enough or too much. Need to point out satellite association. So when you do start marking, even marking your prints today, or when we start karyotyping, satellite association is a phenomenon that we've known for a long, long time. The satellites of the acrocentric chromosomes will orient themselves in the metaphase with satellite to satellite. That is a function of what happened during the previous interphase, that those stalks, actually it's called satellite association, but it's more of a stalk association. And I will show you some staining later on that shows the connection between those two chromosomes. But the ribosomal RNA that's being transcribed, they like to associate with each other during that transcription. And if you catch the cell at just the right time, those are actually still connected by their stalks, but we don't cut them as one because that is a natural phenomenon. It's not a structural abnormality. So, but when you're going through and you're counting this metaphase, it would be very easy to count that as one chromosome. Now, if you did on this one, you would get 45 chromosomes and go, okay, what's missing? But I just wanted you to be aware of the phenomenon called satellite association. The other thing to know about that is that transcription doesn't typically occur on all 10 stalks in the same interphase cell, and we'll talk about cell cycles later as well. Usually, I did some work on this when I was in grad school. Usually I would see in anywhere from 6 to 8 acrocentric chromosomes per cell that had evidence of transcription there. Okay, and then the last slide is just showing how to align the chromosomes on the karyotype page. This is actually a karyotype that was turned into me, and the chromosome 1's lined up very nicely. The centromeres are aligned. Not so much here. Not so much here, not so much here. Now you say, okay, well, those are bent, but there are ways to manipulate them so that we can actually get the centromeres lined up. The reason that's important to a director who's reviewing the case is it's when the centromeres lined up, it is so much easier to compare because you have to do a band by band comparison of both homologs before you can clear that chromosome and say there's nothing structurally wrong with it. So it's just much easier when the centromeres are lined up and the chromosomes are oriented so that the majority of the chromosome is close. See, these are lined up and they oriented it correctly. But if this had been flipped or this had been flipped, horizontally, then you're going to put most of your chromosome, and I can show you sometime the difference. If you put most of your chromosome out here and this is over here, it's too hard to actually do the analysis. This chromosome, they actually did not, this is where ASI trimmed too closely because of the 16 that was sitting here. So the tech should have gone back in and changed those boundaries, it's very easy to do in ASI, and given me a better look at 5P. Extraneous trash needs to be erased. And okay, so flip right 20 and X to position the centromeres as close as possible. So you see, this is a good example of, and we can fairly easily on a 20. Yeah, there are the 3 bands, 1, 2, 3, here's a centromere, everything looks pretty close. But on the longer chromosomes and the higher resolution, this would drive you nuts trying to analyze it. And then this just is a better look and a better analysis tool if this X is flipped. So this way instead of this way. So that is the end of that. And Peng Fei, I think you can stop recording now.